Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is the microbiology of the raw milk. Okay, so the composition of the milk we are already discussed about it, composition and milk processing. Okay, these two topics we are already discussed and the video related to these topics are already uploaded in our check it. Okay, here what should we are going to do? We are going to discuss about the microbiology of the raw milk. We all know that microorganisms are very common. Microorganisms are very common and can easily grow into the food product. Okay, and it cannot be seen by our naked eye. Okay, so for the growth of the microorganism, these factors are important. Fat, Tom. Okay, that means that F is for a food, A is for a acidity, T is for a time, then temperature, then oxygen, and then moisture. Okay, so this we all know that this raw milk are what are highly nutritive. Okay, so this is higher chance that the growth of the microorganism take place. Okay, so when milkman milking this milk at that time, okay, it is pure, means that free from contamination, free from contamination. But as soon as within 30 minutes, this milk will spoil due to the growth of the microorganism. Okay, so here what should we are going to do? We are going to see the how this microorganisms can grow into our raw milk or what is their effect. Okay, now the first one is your introduction. See, milk, but it's very natural, is a natural growth medium for a microorganism. Okay, we all know that this milk are what? Milk are a natural growth medium for the growth of this microorganism. Okay, this normally milk is collected from a lactating animal, means that mostly a dairy cow give a milk. Okay, and milk are collected by those type of animal who are lactating. Okay, at least twice a day and is recognized as a highly perishable foodstuff easily subjected to a microbial contamination. Okay, perishable means that the type of food which can easily spoil, which can easily spoil due to the contamination of microorganism or growth of the microorganisms. Okay, so this is a perishable food and it can be easily subjected to a microbial contamination. This contamination can vary widely due to a milk handling practices. Okay, so this contamination are widely because different handling practices are applied. Suppose that if we take a milk from a milkman, take a milk from a milkman and then we boil it, and after boiling we store it between a 0 to 5 degrees Celsius, then this type of milk will not spoil. Okay, but suppose that we take a milk, boil it and leave it for 12 hours, then it will be spoiled. So the handling practices of this milk will also affect the contamination. It is ranging from milking a few cows by hand in the out of door to milking 3000 cow by a complex. Okay, so there are two type of milking can be done. One is with the hand of the door, with the hand, and it it reaches to the door by a milkman. Okay, and the second one is there is a automated system in a well-equipped parlor. Okay, so the first one is your milking is done with the help of the hand, and the second one is this: milking can be done by a proper automated system which are present in a parlor. In countries with a developing economic, it is not uncommon to find a small quantities of non-refrigerated milk being held by a individual producer to collection center for entry in the market. In contrast, where dairy farming is more advanced technology, milk is refrigerated immediately. Okay, if we talk about the developing country, okay, then in that countries it is not common that the small quantities of the milk can be stored into a refrigerated condition. But if we talk about the advanced technology or a country where dairy farming are in practices, then in that type of countries milk can be stored in a refrigerated immediately after when it is collected and stored in a farm tank until it is picked by a bulk tank driver who delivered the milk to a processing plant. Suppose that milking is done. Okay, now if we have a advanced technology, then what should we do? We refrigerate it immediately after collection, and even it is stored in a farm tank, and it is stored in a refrigerator until it is picked by a bulk tank driver, which transfer the milk to the processing plant. Okay, but in developing country this type of procedure are not followed. They cannot be stored the milk into a refrigerated condition. That's why it will be contaminated by the microorganism. Thus the initial microbiology quality of milk can be anomaly. Nevertheless, there are three basic sources of microbial. Okay, so that's why when the milk are reaches to the industry, first of all they do a microbial analysis, and on the basis of their microbial analysis they accept or reject the milk. Now there are three basic source of the microbial contamination of the milk: from within the udder, from exterior of the test and udder, from the milk handling and storage equipment. So basic these three sources of the microbial contamination in the milk. Milk is at a ambient temperature which is ranging from a sub-zero centigrade. Okay, this milk can be produced at the ambient temperature which are ranging from the 0 centigrade, which is necessary to protect the milk from a freezing. Okay, if we decrease the milk below the 0 degrees Celsius, then it will be freeze. That's why it is important that we have to kept it from 0 degrees Celsius. Okay, and to above 25 degrees Celsius where refrigeration is needed. Furthermore, the duration of the milk storage time on the farm can widely. Therefore, the numbers and the type of the microorganism present when the milk leave the farm can differ, often unpredictably, even under apparently similar condition. Okay, that's why it is unpredictable, because there is a different condition and the growth of the microorganism take place. Okay, now the next one is your: in most dairying areas milk production methods, equipment and on-farm storage have improved over a time. Okay, in a different dairy area, the technique of the milk production, the methods, the equipments, and even the storage area where the milk are stored are improved over a time. However, other disease remain whispered because of the presence of the mastitis associated microorganisms. Okay, this associated microorganisms are already present. That's why the disease remain whispered. Refrigeration on the farm are all too often mask the effect of unsanitary practices like use of inadequate cleaned and sanitized milking equipment. As a result, the microbiological quality of the raw milk supply produced under apparently good sanitary condition, and it can be stored under Adequate refrigeration may be produced off flavor. Yield poor product and present a risk of foodborne infection. Okay, suppose that if the food are if this milk are not stored in a proper refrigerated condition, or it cannot be store or produce into our sanitized equipment, then it is a result that it will produce a off flavor or poor yield, and there is also a chances of the foodborne infection occur into the human being. There are different factors which influence the microbiological quality of the raw milk were reviewed in a bulletin issued by a International Dairy Federation 1980. Okay, so in 1980 there is a review issue, and the name of the review is International Dairy Federation, who describe the factor influence the microbiological quality of the raw milk. Now the next one is your initial microflora of the milk. The number and the types of microorganism in milk immediately after the production, that is the initial flora. Okay, so when milk is produced, then mm, the number and the type of the microorganism are directly reflect the microbial contamination during the production, collection, and handling. Okay, the at that time when milking are done, then the number and the type of the microorganisms present into the milk are directly reflect the microbial contamination during the production, during the collection, and during the handling. If that time the microbial load are low, then the chances of the microbial flora during the production, collection, and handling will also be a low. The microflora in a milk when it leave from the farm is influenced significantly by the storage temperature and elapsed time after the collection. Okay, when a milk is stored at a 14 degrees Celsius, this is low temperature, normally will delay the bacteria multiplication for at least 24 hours. Okay, if we store the milk at 14 degrees Celsius for at least it will delay the growth of the microorganism for a 24 hours. The microflora therefore is similar to that present initially. However, if unsanitary condition exist with the milking equipment or a storage tank, then the low temperature could make these conditions. Okay, suppose that if we maintain the low temperature, but also the unsanitary condition are present, then at that time milk will also be spoiled. Okay, now the next one is, now the useful indicator for monitoring the sanitary condition in a milk uh, present during the production, collection, handling of the raw milk is the total bacterial count, that is called TPC, or standard plate count, that is SPC. Okay, this standard plate count is determined by plating or using a equivalent procedure on a standardized plate count agar, followed by a aerobic incubation for 2 to 3 days at 32 degrees Celsius or 30 degrees Celsius respectively. Okay, what should we do in SPC? For the determining of the microorganism, we play a standardized plate count of agar, and it is followed by a incubation for 2 to 3 days, and this incubation are aerobic incubation, and the temperature it will be a 32 or 30 degrees Celsius respectively. Now the microorganism failing to form a colonies of course will not be the counted. The SPC does not indicate the sources of the bacterial contamination or the identity of the production deficiency leading to a high count. It its sole value is to indicate changes in the production, collection, handling, and storage environment. Follow up the microbial assessment for a psychrotrophs or thermoduric bacteria, spore-forming bacteria, streptococci, coliform can assist in determining of the sanitary deficiency. Now the next one is your types of microorganisms present into a raw milk. Aerobic microorganisms and their respective genera and species that comprise the microflora commonly found in a raw milk which is taken from an individual farm which are located in the United States. Okay, so in from a United States there is an individual farm from which our raw milk are taken. Now in that milk there is a aerobic microorganisms with respect to their genera and species are present. The microorganisms are identified from the isolate from SPC agar following a relatively simple scheme used to characterize these isolate based on morphology, reaction to the gram staining, catalyst production, and formation of acid and gas in a McConkey broth as first described by a Carrera et al. 1955. Okay, so the microorganism then identify or isolate with the help of SPC agar, or you can also identify with the total plate count. Then it is used to characterize and sorry this and this scheme used to characterize these isolate based according to their morphology and reaction to the gram staining, catalyst production, and formation of acid gas in McConkey agar. According to them, we characterize the type of the bacteria, and this was first described by a Carrera et al. In 1955. This scheme is widely used and provide a useful information such as survey data, which are uh, present in a table 2.4 in the next slide. This data is taken from a low count raw milk that it can form around 4000 colliery forming unit which per ml, which indicate that a micrococci and streptococci population dominate the microflora present. Okay, so the total microflora present into a milk in that condition, the micrococci and the streptococci are dominant. In such milk, animal bacteria contamination from the exterior of the udder and from milking equipment is reflected to the presence of those bacteria which are commonly associated with the normal udder and test skin flora. Similar findings are reported in the United States by a donor in 1930. Then this information are shown in figure 9.4 not only it is applied to a low count milk sample from an individual farm, but it also represent the most of the UK form. Okay, so these are the tables. This is what quality incentive program used by a milk cooperative in a United States. The first one is for a quality attribute. Okay, then this is plus 50 premium paid per EVT, 35 premium paid per EVT, no premium, minus 35 premium, minus 50 premium. Okay, now the first quality attribute is a standard plate count. Okay, so in 50 premium paid per EVT, per CVT is 10,000 or below. Okay, now for plus 35, it is 15,000 or below, no premium it is range between 16,000 to 50,000, minus 35 to premium, 51,000 to 1 lakh, and minus 50 premium, it is above the 1 lakh. Now the next one is your pre-incubation count per ml. For plus 50, it is 30,000 and below. For plus 35, it is 50,000 and below. No premium, it is 51,000 to 1 lakh 50,000. Then it is above 1 lakh 50,000 in case of minus 35, and minus 50 it is not applicable. Inhibitory sedimental pad. Okay, in case of premium, no. In case of plus 35, no. No premium, no. And for minus 35, it is not applicable. And for minus 50, it is not applicable. Then added water, no. Okay, maintain grade, somatic count. For plus 50, it is 2 lakh and below. Then it is between 2 lakh 1,000 to 4,000, 4 lakh for the plus 35, 5 lakh 50,000 for no premium, and for 5,000 for minus 35 to 55 for minus 35 for 5 lakh 51,000 to 6 lakh 50,000. Okay, so this is the table for this. Now, the second table is for types of aerobic mesophilic microorganism present in fresh milk which form colonies on SPC agar media. Okay, the first one is your micrococci, streptococci, apogenous gram-positive rod, spore-forming group, GNR group, and miscellaneous group. Okay, micrococci contain micrococcus and streptococcus. Okay, streptococci contain enterococcus group and mastitis, streptococci, Streptococcus aglyceti, Streptococcus digalseti, and Streptococcus uvarius. Okay, Aspergillus gram-positive rods contain Microbacterium, Cornybacterium, Anthrobacter, and Curitia. Spore-forming group contain Bacillus, spore or vegetative cell. GNR group contains Pseudomonas, Acinetobacter, Flavobacterium, Enterobacter, Calabacilla, Aerobacter, Escherichia, Serratia, Algilginus, okay, and miscellaneous group contain Streptomycetes, yeast, and mold. Now the next one is your next table is indicate the incidence of the main group of microorganism in low count raw milk. Okay, the first one is your micrococcus. Incidence is around 30 to 90. 99%, streptococci 0 to 5%, aspergillus gram positive less than 10%, GNR include coliform less than 10%, bacillus spore less than 10%, miscellaneous include streptomycetes less than 10%. Okay, it has been observed that SPC increases the dynamic of the microflora population shift. That is usually an increase in GNRs which are begin to the dominant the microflora at the expense of the micrococci. In most cases, and SPC increases correlate well with unsanitary condition. Okay, when there is an unsanitary condition, then the SPC value will be increases. Okay, now consider the variation of the in the incident of the thermoduric organism and psychotropic in fresh raw milk has been reported. Some differences may be regional or seasonal. Other differences are associated with the methods of the cleaning and disinfecting on farm. Some variation may be accounted by the difference in method of performing laboratory pasteurization for the estimation of the thermoduric count. Time and temperature of incubation of media may account or other variation in both thermoduric and psychotrophic count. Okay. Now the next one is your thermoduric microflora. The genera surviving laboratory pasteurized, which will shown in a table 2.5. Microbacterium, lactinium, and bacteria spores are normally show 100% survival. Some micrococcus genes tolerate can survive. Species of streptococci, lactobacillus, and some cornybacteria are heat resistant. Okay, so when we perform pasteurization during a laboratory, then there are a genera can survive, and this genera can be discussed in a table 2.5. Microbacterium, lactinum, and bacterial spores are normally show the 100% of the survival, and micrococcus can tolerate and can survive. Okay, and the species of the streptococci, that is your Enterobacter faecalis, like lactobacilli and some corony bacteria, also heat resistant and can be survive at 60 degrees Celsius for as long as 20 minutes. But there is a small percentage, an estimate less than 1% normally can survive at 63 degrees Celsius for 30 minutes. Then they will consider as a pasteurization. Okay, most of incidents of the coliform and E. coli found in milk after pasteurization usually can be attributed to a post-pasteurization contamination in the storage and filling step. Okay, if after pasteurization, if the coliform and the E. coli are found in a milk, then this contamination is called a post pasteurization contamination, and this contamination are occurred during a storage and filling step. However, one must go back to the early work of the Olsen, Mackay, and Advorson 19.5 to appreciate the thermal resistance of the E. coli. These researchers reported that the growth condition exists in collecting and cooling of raw milk could influence the thermal resistance of the bacterium. Okay, thus the Z value was introduced and adjustment to the pasteurization temperature above 63 degrees Celsius were made to assure a advocate destruction of the E. coli. Okay, so there is a research which said that there is a chance that E. coli can grow at the range of 63 degrees Celsius, 63 degree Celsius temperature. Now, that's why the pasteurization temperature will increase above the 60 degree Celsius for the advocate destruction of the E. coli. In contrast, the bacillus spores contained of raw milk rarely exceed around 5000 ml per ml 5000 per ml, and the bacillus spore generally is present in higher number during the winter than in summer. Okay, then there is this bacillus spores are generally present higher in number during the winter as compared to the summer. This is because these bacteria are dominant milk microflora during the winter and derive largely from the surface of the test that have been in contact with the bedding material and soil associated with housing of the dairy herd. Okay, this table is for our thermoduric and psychotrophic microorganism present in a raw milk. That is your microbacterium, micrococcus, bacillus spore, clostridium spores, and alkalinus. Now, Ridgway found that around 12% of the farm milk supply from winter house dairy hubs demonstrate spore count of less than 100 per ml, and the count were much lower in summer months. Bacillus licheniformis is the most common species which present, while Bacillus cereus is found only in a sporadically in a bulk tank milk. Milk can are known to be a primary source of the Bacillus cereus spore in a milk. In contrast to the spores, micrococci and microbacterium species are also derived almost exclusively from a milking equipment that is sometimes so heavily contaminated with this type of bacteria that thermoduric count in the milk often exceed 5 into 104 RNF. Most of the thermoduric organisms do not multiply applicably in raw milk even at a ambient temperature. Okay, thus during a high thermodynamic count in milk which are held for a 24-hour could be reliable evidence of the gross contamination from the milking element equipment. For this reason, the thermodynamic or laboratory pasteurization count has been proposed in the United Kingdom. However, it is reported that there is a weak correlation between the standard plate count and the thermodynamic count when it is used as an indicator for poorly clean milking equipment. The next one is your psychotrophic microflora. Okay, psychotrophic microflora are those microorganisms that can thrive under a refrigerated temperature, that is the temperature range between 0 to 5 degrees Celsius. Okay, but it can be grow between 3 to 7 degrees Celsius, and it can be excellent review by a psychotroph found in milk and dairy product which are given by a cuisine in 1982. Okay, in 1982 cuisine give a milk psychotrophs found in milk and milk product review. Thus, commonly occurring psychotrophs in raw milk are GNRS, which will be discussed in table 2.5. Pseudomonas species account for at least 50% of the GNRS, with a Pseudomonas jolorescens predominating. Okay, then other species also present like your Pseudomonas putoidea, Pseudomonas fragi, Pseudomonas aeruginosa, Flavobacterium, Acetobacter, Moraxilla, Acrobacter, alkali genes, Chromobacterium, Aromonas calabacilla, and the coliform group, which are comprise most of the remaining psychotrophic GNRS. Okay, some of these psychotrophs, when growing in a refrigerated milk, it produce extracellular heat stable lipase that contribute to the species their can development of the rancid flavor and well proteinase and degrade the KC. Even among strain of one species, there can be a considerable variation in activities of Pseudomonas LL ILL0 reasons, which in most cases produce both lipolytic casinolytic enzyme. The gram-negative cytocops are destroyed during the pasteurization, but their enzyme remain active. As a consequence, these bacteria are of considerable importance in manufacturing milk and milk product, and are most commonly associated with the post of the pasteurization, which are given by a Philip ETL 1982 and 1981 and Cuisine 1982. The next Incident of the psychotropic incidents stain by a bacillus species that contain spores in an individual producing milk is low, but the seldom exceeding 10 m per ml. Those species found in a raw milk, including B. coagulate, B. circulus, and B. cereus, B. pumilus, and B. subtitles, Anthrobacteria, and other gram-positive species also have been reported as associated with raw milk psychotropic microflora. The psychotropic microflora is derived from our teat surfaces as poorly defined, relatively inactive group that include microbial spores like Corynebacteria and Giardiasis, indicate cleaning and disinfecting of milking equipment continue to be the main reason for the psychotropic Giardiasis in a raw milk. Okay, now the next one is your coliform bacteria. Incident related to this coliform belong to the genera Enterobacter and E. coli in a raw milk and have been received considerable attention. This attention is partially because of their association with the contamination from a fecal origin. Okay, because this coliform contamination are occurred due to the fecal origin and the consequent risk of other pathogenic fecal associated microorganism being present, and partially because of the spoilage of their growth in milk, and can also produce due at a ambient temperature. It now well recognized that the presence of coliform in raw milk is not evidence of direct fecal contamination and should not be relied upon detected in advocate udder cleaning prior milking. Chloroform can rapidly build up in moist milky residue like your biofilms and milking on milking equipment, and then become a major source for the contamination of the milk being collected. Okay, this coliform are grow in a biofilm which is moist and milky residue. Okay, it not contaminated by the fecal okay, fecal material, but it is grow into a moist and milky residue of biofilm on milking equipment. Thus it become a major source for the contamination. However, relatively low coliform count in milk do not necessarily indicate effective cleaning and disinfectant of the equipment. Coliform count regularly in excess of 100 per ml, okay, are considered by some sanitation to be evidence of unsatisfactory production practices leading the environmental contamination of the milk. Okay, if milk contain a 100 coliform in per ml, then it will consider as a unsanitary satisfactory production practices and leading to a environmental contamination of milk. So this is all about the microbiology of the raw milk. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platform like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe.